DNA sequencing requires a single stranded template, and obviously we need multiple copies of that. So in order to do that, initially scientists, they started using bacteriophage M13 that was actually engineered to give us this single stranded templates. It's a rod shaped virus that is actually itself, it contains the single stranded DNA, and this single stranded DNA then gets converted into the double stranded DNA while it's there in the host. So this is how we can see its life cycle. Initially it enters into the bacteria. Since it's a bacteriophage virus, so it attacks bacteria. So here is we see that it's a single stranded DNA, and then it gets converted into double stranded form, and then this double stranded form gets replicated. So we get lot of double stranded forms, and then after that they give rise to lot of single stranded forms, and then these single stranded forms, they packed into after the gene expression, and they give their coats or proteins, and then they get into those proteins and packed into virus particles and leaves the cell. So we can engineer this virus to have our single templates. So M13 has been engineered to contain multiple cloning site that is actually contained within the N-terminal site of LAC-Z gene. So in this way we can also do the screening. Normally the template is cloned into this double-stranded form, RF replicative form, and while we put our gene of interest, since it's there in the LAC-Z gene region, so it knocks out the LAC-Z gene, and obviously there is no production of the beta-galactosidase, which in normal cases it interacts with the X-gal and gives the blue color, but knocking out of that gene will not give us that blue color. So in this way we can screen that our gene is there or it's not there. So here is the gene for which we want to make the template, the DNA. We put it there into the multiple cloning site of this M13 vector, and then obviously once it's landed over there, we can also design the primers of the neighboring regions since we know this whole sequence. So it's easy to design the primers, and then we can have the multiple copies of this single stranded templates coming out, which we can use for our sequencing. Cloning of these genes into the vectors that are kind of complicated procedures. So PCR might be taken as an efficient procedure, but there is one drawback that in PCR it's really it's need lot of skill to develop the good primers. So obviously cloning into the cloning vectors, they might have their own benefits also. Same way PCR is more efficient as compared to them. So in the end we conclude that the various discoveries in the molecular biology that has helped in the evolution of the genome sequencing technologies.